Hi, Mr. Sheehan here, and this series of slides we're still on blood, and these slides will be on hemopoiesis and turnover, primarily of red blood cells. So the opening slide just shows an erythrocyte, red blood cell, white blood cell, and a platelet. This slide is not to be memorized. It's out of your textbook, OpenStax figure 18.4 in this chapter. And what I want to have you take a look at, let me move this bar out of the way again. Is just basically the flow of cell development from stem cells. So again, you don't need to memorize this, but just sort of follow along, look at it, and see if you can make sense out of what the diagram is trying to show. So it starts up here at the top with stem cells, and these are referred to as hematopoietic stem cells, so they're blood stem cells. And the multipotent means that this stem cell, can differentiate into other types of blood cells, multiple blood cells, but it can't develop into hair cells, bone cells, muscle cells, etc. So these stem cells are limited to blood cells in their formation. So the idea is that you start with the stem cell and it goes through mitosis and you wind up with two daughter cells. And one of those daughter cells will just remain a stem cell. So it can go back and do the same thing. The other cell will begin to differentiate, and it can go into one of two paths. They can't back up. That's just the way stem cells are. So the cell, when it divides, it could go to the lymphoid stem cells and follow this path, or it could go to the myeloid on the left. But let's just follow the path on the right for a moment. Again, I'm not asking you to remember the different types of cells, but lymphoid, lymph is referring to the lymph system, of course, lymph nodes, etc., where many of these cells reside. And if you remember from A&P1, blast usually means immature cell. So in this case, a lymphoblast is incomplete in its development. It can go on to form what's referred to as a natural killer cell, which is a type of white blood cell that we'll discuss very briefly in the immune system. The other type of cell it can develop into are lymphocytes. And the two important types of lymphocytes to remember are that there are T lymphocytes and there are B lymphocytes. And we will be talking about those more in the immune system. So, generate white blood cells in the bone marrow. And to back up to the top of the figure, taking the other path, myeloid is referring to bone marrow in particular. And the myeloid stem cells can differentiate into four different lineages, four different types. And again, all I really want you to pay attention to at this point are the different types of cells that get formed. So we have lymphocytes here that come in two flavors. We have monocytes, eosinophils, neutrophils, and basophils. And all of these are white blood cells. They're all part of the immune system. All of those. The other cells that can form back up to the top, you can go down this pathway and we have a proerythroblast, and then we have a reticulocyte. And I would like you to remember reticulocytes They are immature red blood cells, so when they are complete their development, they will be an erythrocyte or an RBC. And the other sort of formed element in the blood is on the far left. You have a megakaryoblast and then a megakaryocyte. But what's important, let me move the screen up a bit, are the platelets. So this is where platelets are formed. And they form from a megakaryocyte, and they are essentially just extensions, little bits of the cytoplasm. They get pinched off, they like bud off, and then that's what the platelets are. So they're little bags of cytoplasm from this much larger cell that is residing in the bone marrow. So that forms platelets. So platelets, erythrocytes, leukocytes, and that's pretty much it for that slide. So you should be able to just kind of follow along with it, make sense out of it, recognize the types of cells at the bottom of the figure that are formed. So this slide just introduces the different types of white blood cells or leukocytes. And we'll go over these in more detail when we get to the immune system later. So, neutrophils. They are phagocytic. That would be one of their main claims to fame, is that they phagocytose bacteria, worn out cells, debris, garbage that they come across, and they're sort of the cleanup crew. And what's noteworthy about a neutrophil is that the nucleus is rather bizarrely shaped. It sort of looks like badly formed sausage. You get 3 to 4 links, they sort of kink in different places, and the nucleus is highly variable. So that's one of the criteria for identifying a neutrophil is the nucleus. And the lymphocytes that we just looked at, this is what they sort of look like. If we had a lab, you'd be looking at these cells underneath the microscope. This is a RBC for size comparison. They're about the same size. And lymphocytes, the nucleus, pretty much fills all of the cytoplasm. Monocytes will develop into macrophages. Again, we'll talk about this later. These cell types, macrophages also phagocytose bacteria, etc. And then we have eosinophils, which we won't say too much about, and basophils. The thing to note about basophils and the thing to remember about basophils is that they release histamine. And basophils, as we'll look at later when we get to the immune system, the basophils can develop into mast cells. Very similar. One's floating in the blood, one's out in the tissue. So that's not most. Let me start over here. So basophils can develop into mast cells. But both mast cells and basophils release histamine. That's important. You ought to remember that, because we will be doing a fair amount with histamine and the immune system when we get to that. So, this cell, or this slide, is sort of an introduction to, or just more comments rather, on the development of red blood cells. So you know what a reticulocyte is. It was an immature red blood cell. It is ejected, the nucleus is ejected while the reticulocyte is still in the bone marrow. So that's where all cells begin their all blood cells begin their development, is in the red bone marrow. Reticulocytes that are going to become mature red blood cells, they eject their nucleus, and this is pretty much what they look like as a reticulocyte. They exit the bone marrow and then they circulate in the blood for a short period of time. So these three are the reticulocytes, and they form sort of this, they have a reticulum, or sort of a network, strands or filaments. So those are the dark strands of reticulum. They're clumps of ribosomal RNA and other organelles that have not yet been ejected. So remember, cells start out with all the organelles. They have a nucleus, the ribosomal endoplasmic reticulum, the rough endoplasmic reticulum, smooth endoplasmic reticulum, the Golgi body. All that machinery gets ejected from the red blood cells. So when they're circulating in the bloodstream, red blood cells primarily just contain hemoglobin. They don't contain mitochondria. So they circulate for about a day before finishing their development into an erythrocyte. And the number of reticulocytes is usually about 1 to 2%, not a lot. And the amount of circulating reticulocytes is the retic count. So a retic count is literally counting the number of reticulocytes in a volume of blood and coming up with a number. If the retic count is low, it could indicate that there's a problem in forming new red blood cells sufficiently. If the retic count is too high, it could portend problems with the bone marrow, possibly a tumor, a blood cancer, where you wind up with polycythemia. So they need to be within a certain level. But that is a retic count. So some notes on red blood cells. Again, they circulate for approximately 4 months, then they wear out. No nucleus, so they can't do any protein synthesis. They're packed with hemoglobin, and this is the abbreviation that we're going to be using for hemoglobin throughout. It's quicker to write, quicker for you to take notes. And hemoglobin its function is to carry oxygen, molecular oxygen, O2. There are no mitochondria, as I mentioned, so they are incapable of aerobic respiration. They can do glycolysis only. And mention that red blood cells are packed with hemoglobin. So I think this picture is out of your textbook. So this is one molecule of hemoglobin shown. There's a single molecule. It's made up of 4 individual polypeptide chains. There are 2 alpha chains and 2 beta chains that you don't really need to remember, other than that there are 4 polypeptides. But there are 2 betas. There's a beta 1, beta 2, and there's an alpha 1 and an alpha 2. Okay. Each one of these polypeptides contains a non-protein heme group. So there's a red polypeptide, a pink, a yellow, brown, etc. And each one of those has a heme group in it. So in each molecule of hemoglobin, there are 4 heme groups shown on the right. Here is the heme. It's not a protein. It's a complex ring structure with an iron in the center. And it's that iron atom that can bind and release molecular oxygen. So when we look at the breakdown of red blood cells, we're going to have to look at the heme portion. We're going to look at the globin portion, the peptide, the non-protein part, and the iron. Let's take a look at this slide for a moment, and it is showing some difference between fetal hemoglobin and adult hemoglobin. So the site of synthesis is different in the fetus and the adult. So I put this figure in the slide, not for you to memorize, but for you to practice interpreting. So what I would like you to do is be able to look at slides and just figure out what the story is that it's trying to tell you. Don't worry about trying to memorize it. Just focus on interpreting it, maybe explaining it to your mother or your sister or somebody else. What does this figure mean? So when you do that, just start by looking at what the axes show. And on the left, it's the prenatal life and months. So this is fetal. This is in the fetus, in utero. And here is birth. So there's a dividing point. So 3, 6, 9 months and then birth. And then after birth or postnatal life and months. So a 6-month-old baby. And the bottom half of the figure is showing the total hemoglobin of 100%. And what it shows is that there are 2 different types of hemoglobin. There's a fetal hemoglobin and an embryonic hemoglobin. And as the embryonic hemoglobin is this dotted line, very short period of time during embryonic development, and then the fetal development begins, and then you have the red line. So what the figure shows is that during most of the prenatal life, most of the fetal development, it is fetal hemoglobin that is in the fetus's red blood cells. And slowly the amount of adult hemoglobin begins to increase. So you have several types of hemoglobin in the red blood cells of the fetus. Primarily fetal hemoglobin. And then after birth, the fetal hemoglobin declines rather sharply, and the adult hemoglobin picks up. And that's what you and me and everybody else, all the other adults, are forming, is adult hemoglobin in the red blood cells. The upper half of the figure shows the site of erythropoiesis. So where in the body is the hemoglobin being synthesized? Well, very early on, it's in the yolk sac during the embryo. Later, in early fetal development, it's the liver that forms the red blood cells. The spleen kicks in for a little bit, and then the liver starts to pick up, or the bone marrow, rather. So in the adults, all of the erythropoiesis is done in the red bone marrow, and that is where the hemoglobin is synthesized. So, one of the things to note that we'll talk about more when we do reproduction is that the fetal hemoglobin needs to steal oxygen from mom's hemoglobin in the placenta. So the 2 bloods don't mix in utero during development. Fetal hemoglobin stays in the fetal circulation. The maternal hemoglobin stays in the maternal circulation, and they do not mix. The bloods don't mix. They stay separated at the placenta, where gases and nutrients and wastes can go back and forth. But the cells don't mix. So fetus has to get all of its oxygen from mom's blood. So mom's oxygenated blood comes to the placenta, carried on adult hemoglobin. Baby's blood comes to the placenta, and it's carrying fetal hemoglobin. The two hemoglobins are side by side, and the baby's hemoglobin, the fetus, needs to get oxygen from mom. So the fetal hemoglobin has a higher attraction for the oxygen. So it basically tears it off of mom, binds it, and then goes back off to the fetal circulation to supply oxygen to the developing fetus. We'll do a little bit more of that when we get to reproduction, but that's the basic idea. This figure is looking at erythropoiesis, or formation of erythrocytes. And we'll sort of develop it into a feedback loop. So this is in the adult, and the amount of erythropoiesis is regulated. So this on the left is our basic picture of homeostasis. The idea is that here is the oxygen delivery to tissues, the amount of oxygen that can be delivered, and then time, of course. And the idea with this model of homeostasis is that you have a high limit and a low limit, however you want to define those, whatever numbers you want to put on that. But an upper limit and a lower limit. And normally the red blood cells, or the oxygen delivery capability, sort of stays within this normal boundary. That's our model of homeostasis, a range of homeostasis. If the oxygen delivery to the tissues drops too low, down in this area, then we have stress, physiologic stress, where it's low, and that is hypoxia, where you are unable to deliver enough oxygen to the tissues, hypoxia. That's stress, and that is detected by the kidneys. So blood flows through the kidneys, of course. It's about 20% of cardiac output flows through the kidneys. And the kidneys are monitoring the amount of oxygen in the blood. And when it goes low, such as with hypoxia, the kidneys detect that. And then the kidneys will release a hormone that's abbreviated as EPO. And EPO stands for erythropoietin. So erythropoietin is a hormone. It circulates in the blood. It goes to the bone marrow, and it stimulates stem cells, which will ultimately make more red blood cells to fix the problem of too little oxygen carrying in the blood. But let's consider some possible causes of hypoxia, which was our original stress. The patient could have suffered from hemorrhaging or loss of blood. So if you spill out erythrocytes, blood cells, they're not in the blood to carry the oxygen. You're going to need some more. The patient could be suffering from some type of anemia, of which there are multiple causes. Or the patient could be suffering from high altitude effects. So the basic relationship is that as you go up in altitude, up in elevation, the amount of oxygen that's available decreases. So in Yakima, we're about 1,000 feet or so above sea level. But if you drive up to Paradise on Mount Rainier, that's at about 6,000 feet. If you run up the hill on Rainier for a ways, you're out of breath. Or you fly from, let's say you live in Seattle, you fly to Denver, as I did one year. You get out at 5,000 feet, you jump in a car, you drive up to somebody's house, and they're at 8,000 feet. That's where they live. And you go running up the stairs, and you're out of breath, and you go, gee, this is strange. Well, your blood is slightly hypoxic. You stay there long enough, you'll acclimate to it. And here we go. Kidneys releasing EPO. Targets the stem cells. And we've already been through this. The stem cells then go through this process. This is erythropoiesis. And they form, reticulocytes are formed. And then the reticulocytes develop into mature erythrocytes. So we would wind up with more reticulocytes. We'd wind up with more erythrocytes, red blood cells, and that would lead to increased oxygen carrying capacity of the blood. So I'd like you to understand the basic feedback loop of erythropoiesis and the role of EPO. And there are some questions in the objectives concerning EPO. Oh, this is practice. So what I would like you to do is to stop the video for a couple of minutes and then practice what we just went through. So what I would like you to be able to do from memory is to draw that negative feedback loop that we just went through on your own, on a blank piece of paper, and see if you can recreate it. So start with the stress of hypoxia, and then work through this feedback control process to correct stress. Complete that loop. So I'm going to pause the video, or you pause the video for a moment, and try that. When you get stuck or when you want to check your work, come back and we'll complete it on the screen here. Okay, you're back. Let's work through this together. Hopefully, you were able to get a good start on this and complete it on your own. I would practice this again. So let's just work through the loop. Start with the stress and define the stress. And our stress is low oxygen in the blood. And then somebody needs to detect that. So those are the kidneys. Kidneys detect. And the kidneys release EPO into the bloodstream. EPO is a hormone. And EPO then targets stem cells in the red bone marrow. That stimulates mitosis, and we wind up with more red blood cells. We wind up with an increased retic count. The number of circulating reticulocytes, those develop into more red blood cells. And with more red blood cells, then we can carry more oxygen in the blood. And that fixes our stress. And so we sort of completed the loop by going from stress to fixing the stress. So, closing the reflex loop. Next slide is taking a look at the breakdown of red blood cells. So what we've done so far is to look at the synthesis of red blood cells. We just finished that. So this slide is a little bit busy. Don't panic trying to memorize the whole thing. Again, let's just work through it. And what I would like you to do is to be able to interpret it. And just follow along. It's not bad if you just take it one step at a time. So let's figure out where to start. And let's start over here. Oops, that's going to be off the screen. So we will start here as we work our way through the figure. So, red bone marrow, red blood cell formation, reticulocytes, leave. Now we're out in the circulatory system as a newly made red blood cell, released into the circulation. They circulate for about 120 days, about 4 months, and then they wear out. Remember, they can't make protein, they have no nucleus, but they will eventually wear out, and then they have to be recycled, which is what this slide is looking at. So the red blood cells are then endocytosed in a macrophage, and these macrophages are in the spleen, in the liver, and in the bone marrow. So those are a white blood cell, and they gobble up the worn out red blood cells, and then they break them down. So here's the breakdown of those components of the red blood cell. So they're filled primarily with hemoglobin. That's the primary protein. There's not much else inside a red blood cell. So you break down the globin portion, the beta chains, the alpha chains, and you release the amino acids. And the amino acids then go back into the metabolic pool, if you will, and you can reuse those amino acids to either make more hemoglobin or make hair cells, hair protein, or bone protein, or anything else. So that takes care of the globin, the amino acid side. The heme portion, remember, has two pieces, the non-protein and the iron. So if we follow the iron first, you separate that from the heme, and the iron gets transported through the blood. It's not terribly soluble, so it's bound to a transferring protein called transferrin. And transferrin is the protein that shuttles iron through the blood, and that can go back to the red bone marrow for synthesis of new hemoglobin, or it can go off to the liver and be stored there for later use. So that takes care of the iron. The non-protein part of the heme, that ring, takes this path. It gets converted into several intermediates, one of which is biliverdin, which we won't worry about, but then we will follow bilirubin. And bilirubin is an important molecule that we will be following in just a moment. Should have remembered that was going to go off. So let's follow the bilirubin. Remember what it is. It's that non-protein part of the heme. Well, it goes back into the bloodstream. Let me change color back to red for blood. The bilirubin goes back into the bloodstream where it circulates through the liver. And then in the liver, if the liver is functioning properly, it will remove the bilirubin from the blood. And one of the ways that it can do that is that it will take the bilirubin and excrete it into the bile. So it's stored in the gallbladder that we'll come back to with the digestive system, but then it gets released as bilirubin into the small intestine, at about the place where the pancreas empties. And that then flows through the small intestine. It goes into the large intestine. Let me move the slide up a bit. So the bilirubin then goes through the large intestine where it gets broken down into several other intermediates, urobilins, serocobalamin, etc., that I'm not going to worry about, and then it gets eliminated in the feces. So that's a major way of getting rid of the bilirubin, and it's primarily what gives feces all the same color. No matter what you eat, it pretty much comes out the same color, primarily due to the bilirubin breakdown in the large intestine. So that's one way of getting rid of the bilirubin. Another way is that in the intestines, a lot of the fluid gets reabsorbed. So bilirubin may get reabsorbed back with blood, and then it goes back to, circulates in the blood and goes to the kidneys. Likewise, not all of the bilirubin that came to the liver is sent through the bile. Some of it goes back into the blood and goes to the kidneys. So the kidneys too can take bilirubin, break it down, and then eliminate it in the urine. And that's pretty much what gives urine its common color of yellow, is the breakdown of the bilirubin. So I'd like you to follow along with the recycling of the protein, the amino acids, the iron, and the heme. And the heme is converted to bilirubin, which circulates through the blood. It's cleared from the blood by the liver and the kidneys. From the liver, it goes into the bile and out with the feces. From the kidneys, it's cleared through the urine to the toilet. This figure shows a couple of pictures of jaundice. And the photos are actually about true color. The one on the upper right is a patient with severe jaundice, and they can actually look yellow-green. When you look at them, you go, oh my gosh, that person is sick. They are yellow-greenish. And another place that jaundice shows up is in the lighter tissues, part of which are the sclera, the white of the eye. It is a common place to see severe jaundice. So jaundice is the result of excess bilirubin in the blood. That's the definition of jaundice, is the coloring. See yellow-greenish coloring to the skin or the sclera. Some common causes for jaundice, they generally involve liver compromise, liver damage, hepatitis, which is inflammation of the liver, either acute or chronic, And if you're not clear on the difference between acute or chronic, look it up, make a flashcard. But acute comes on, quickly disappears typically. Chronic is long-term, there for a long period of time. Cirrhosis of the liver from alcoholism, common risk of heavy drinking is damage to the liver. Liver cancer from various different causes, and then drugs or other toxins which can damage the liver. So the liver isn't able to clear that bilirubin from the blood. And the last slide here is a brief discussion of neonatal jaundice. So this is jaundicing of the neonate, or the newborn, the baby. So the idea is that, take a look at the figure on the right again. This should look familiar. You ought to go back perhaps and see previous figure, which was slide number 24. And compare. They're pretty similar. So it shows the after birth, the demise of the fetal hemoglobin, and that's what we're going to be paying attention to for the jaundice, and the rise of the adult hemoglobin. So it's this clearing of the fetal hemoglobin, that it gets broken down, releases all the bilirubin back into the blood, causing the jaundice. So it's a transition from fetal hemoglobin to adult hemoglobin, and the fetal hemoglobin is broken down, similar to the slide that we just saw. However, in the neonate, in the newborn, the liver isn't functioning 100%, so it has difficulty clearing that bilirubin load from the blood, so it builds up in the skin. And a vitamin K shot is typically given, it may be routine on newborns, to help stimulate the liver. Kind of kickstarts it, gets it going, wakes it up to help clear the bilirubin. The danger of excess bilirubin in the blood is that it can become toxic. It's a neurotoxin. If the levels become elevated, it can cause brain damage. So you want to watch for that. So these days, when a newborn is suffering from neonatal jaundice, they'll put them in a little tanning booth in the neonatal unit and put glasses on them and put them under UV light. And UV light has enough energy to penetrate the thin layers of the skin and break down the bilirubin that's passing under the skin in the capillaries. So it helps to break down the bilirubin, clear it from the body so that it's no longer toxic. In the old days, if you will, before we had neonatal units, or in other parts of the country where they're just not, or the world where they're just not available, you would just take the baby out, put them on a blanket out in the sun for a short period of time each day, and the sun, the UV from the sun, would break down the bilirubin and do the same thing. So, review those slides. If you have questions, let me know.